This next series of podcasts is going to deal with cytogenetics, and I'm going to show you a list of learning objectives for the next several podcasts. But before I do that, let's just consider this diagram that represents two human cells. The big one is an example of a neuron, and the little red dot would be an example of a lymphocyte. These two human cells have identical genetic makeup, that is, they have the same genotype. So, question to keep in the back of your mind: how can cells that have the same genotype exhibit such vastly different phenotypes? Learning objectives for the next few podcasts. You should be able to describe the structural organization and packing of DNA into chromosomes. You should be able to explain the differences between chromatin and chromosomes. You should be able to explain the differences between heterochromatin and euchromatin, be able to give examples of each of these. You should be able to explain the function of the nucleolus in ribosome biogenesis and be able to identify the NOR chromosomes in humans. The NOR chromosomes would stand for the nucleolar organizer chromosomes. And you should be able to explain how molecular genetic processes such as transcription, replication, recombination would relate to chromatin and chromosome structure. Here is an example of a set of images that look at levels of chromatin packing from a native DNA molecule to a metaphase chromosome. And you should be familiar with this from your studies of biochemistry. So you can look at DNA existing as a naked DNA double helix. You of course know that the DNA double helix associates with nucleosomes. The nucleosomes pack into the basic chromatin fiber, the so-called 30 nanometer chromatin fiber. That fiber continues to pack and condense until you get a fully condensed metaphase chromosome. You might imagine that as this chromatin fiber packs, you might have DNA sequences that may be many base pairs apart from each other, but as the chromatin fiber packs together, these DNA sequences may be brought closer together in three-dimensional space. Just to remind you that nucleosomes consist of a protein core of histones and DNA. The histones would be a histone octamer that consists of 2 moles each of the histones 2A, 2B, histones 3, and histone 4. On average, about 200 base pairs of DNA. There's perhaps 140 to 160 base pairs in the nucleosome core region. That has approximately 1.65 turns of the double helix in a left-handed coil. The linker region, about 40 to 60 base pairs of DNA. You should maybe recall from your studies in biochemistry that the diploid human genome, perhaps 6.4 times 10 to the 9th base pairs of DNA, that's about 30 million nucleosomes. You might remember that histone H1 binds to the linker region and to the core particles. The bottom image here is prepared under non-physiologic conditions. It shows the nucleosomes as such, the so-called beads on a string appearance. So the core region would be like that, or like that, with the linker region in between. The top image is under physiologic conditions. This would represent the 30 nanometer chromatin fiber. This would be what the chromatin fiber would look like in an interphase cell. As you might imagine, the 30 nanometer chromatin fiber can pack together or unpack depending on the state of genetic activity or gene expression. Modifications to histones play crucial roles in not only packing of DNA into chromatin, but in also making DNA sequences available for replication, for repair, for recombination, and for transcription. Modifications in DNA itself are also important in regulating gene activity, and you should have a reasonable appreciation of this from your studies of biochemistry. As you might remember, histone modifications occur at the N terminal end, and these modifications may explain how DNA can relax. The diagram shows the DNA packing into the nucleosomes and ultimately into the 30 nanometer chromatin fiber and eventually compacting into a metaphase chromosome. The diagrams on the right show a space-filling model that is actually based on X-ray diffraction studies of nucleosome particles, with the typical ribbon diagram that's commonly generated by computer of complex protein molecules. So you can see the red are the H2A molecules, the kind of the orange the H2B, green showing the H3, and the blue showing the H4. The gray areas in this diagram show where covalent modifications to histones might occur, and also note that the N-terminal ends of the histones stick out from the main center of the molecule. As you understand, DNA can wrap around this histone octamer, and that wrapping of DNA may be influenced by how the histone tails might be modified. And so you might imagine that modification of the histone tails can allow DNA binding proteins to remodel the nucleosomes within the 30 nanometer fiber, and that may be very important as one considers molecular genetic processes like transcription or like replication. So the cartoon might show here the histone molecules with a remodeling complex allowing DNA to spin a little bit loose from the histones so that these sequences might be available for molecular genetic events. You could have molecular genetic events occurring, then a remodeling complex where you dissociate DNA binding proteins, and the DNA might then reassociate with the nucleosome core particles. We also talk about epigenetic regulation of transcription, and this table looks at some general principles which you need to understand. That epigenetics relates to heritable changes in gene activity that are not caused by changes in DNA sequences. So, for example, you may methylate GC-rich regions in DNA. If these GC-rich regions are unmethylated, the genes there may be active. If they're methylated, the genes may be inactive. Also, you might acetylate histones. Acetylated histones suggest that genes may be active, whereas deacetylated histones suggests that genes would be inactive. As we said, epigenetic inheritance relates to modifications to histones and to non-histone chromosomal proteins and potentially to the DNA itself, but it's not changing the nucleotide sequence. And this is a hypothetical example to demonstrate regulation of gene activity in the development of the nervous system. So, for example, the chart looks at a lifespan, embryonic life through fetal life, infancy, childhood, all the way to adolescence. And it might look at some significant types of genes that might be expressed. You might, for example, expect that genes for cell proliferation are going to be expressed very early on in embryonic development, and they're going to decline through fetal life and infancy, and of course will drop to lower levels in post-fetal life. You might look at genes that are involved in the development of the nervous system, like, for example, dendrite development or myelination, and you may see lower expression of these genes earlier in the lifespan, but then the expression of these genes certainly would get much higher as the lifespan continues. And this is just an example to demonstrate regulation of gene activity, in this case for the nervous system, but it's just to indicate that gene activity is regulated in a time-dependent manner during an individual's lifespan. This diagram shows a simplified mitotic cycle, so it would be looking at somatic cells, and if you try to relate what's going on with the DNA and chromatin during a cell cycle, so interphase consists of G1 and G2, DNA replication, repair, and recombination, RNA transcription, splicing, translation, and modification. All of this would occur during interphase. During the cell division cycle, of course, the chromosomes segregate. In somatic cells, you have one cell division, amitosis. In gametes, there are two cell divisions, the mitotic divisions. The cartoon indicates the state of the chromatin at interphase at G1 as DNA is replicated. Then you see the chromatin at G2. And of course the chromatin gets highly compacted during the division cycle, and during the division cycle all of these molecular genetic events would be halted. We know that DNA exists in the configuration of chromosomes at the M phase, that is at the cell division phase, but the DNA exists as chromatin at interphase. As we've suggested, replication, transcription, and translation stop during the M phase, and of course during the M phase the chromosomes are captured by the spindle.